Morning, folks. So a couple things. We have an exam coming on the 25th, and I wanted to remind you that I'll be holding a special review session on Friday night. That's February 22nd, Winnell Hall, 7 to 9 p.m. This will be posted on BSpace later, but I just want to make sure that everyone's got that cranked into their calendar in case you want to come. The other thing is that this week, for office hours, if you're looking for me this week, on Friday, I'm going to hold office hours 12:15 to 1:15 in the usual room in DLSB. Okay, 12:15, 1:15 Friday of this week. And of course, today, the usual time is 12 to 1, same room for office hours for me. Good. So this week, we're going to talk about energetics, chapter 9 in your book. I want to get into that today, but I first want to just quickly recap a couple of things about enzyme inhibitors that we were talking about at the end of class on Friday. So just as a quick reminder, we talked about the fact that we can categorize enzyme inhibitors into classes based on their behavior and to some extent based on their chemical structure, right? So we talked about competitive inhibitors. These are molecules that bind directly to the substrate binding site in an enzyme, and they tend to look a lot like the substrate itself. They have a chemical structure similar to the substrate, and so they can directly compete for binding. And in contrast to competitive inhibition, we also talked about non-competitive inhibition. And these kinds of molecules typically don't look like the substrate molecule. Instead, they're designed to bind to a site elsewhere on the enzyme. And importantly, they can bind to the enzyme whether or not a substrate molecule is bound, typically, for non-competitive inhibitors. And then a third type of inhibitor is an uncompetitive inhibitor. And these are kind of a specialized category of molecules that only bind to the enzyme substrate complex itself. So again, they're molecules that typically don't bind to the active site or to the substrate binding site per se. They bind somewhere else, and they only bind to the enzyme substrate complex. So this might be a situation where when the enzyme binds to the substrate form a complex, there's some sort of conformational change that now enables this type of inhibitor to bind to it. And when that happens, the enzyme is now not competent to catalyze reactions, or at least not as well. Okay? And very importantly, we can determine the type of inhibition by measuring enzyme kinetics, meaning the rate at which the enzyme is catalyzing reaction with different concentrations of substrate as a function of the concentration of the inhibitor. And this is something that if you take biochemistry, if you take MCD 102 or 110, for example, you'll get into this in a lot more gory detail, okay, about how you actually do that. But I want you to be aware that you can actually figure out a lot about the type of inhibition by simply measuring how the rate of the reaction is affected in the presence of different concentrations of the inhibitor. And depending on the type of inhibition, the kinetics will be affected differently. Okay? So we also talked towards the end of class last time about allosteric effectors. These are molecules that also bind to a site, typically not the active site of an enzyme. And very typically, if we're talking about an allosteric effector molecule, we're talking about it in the context of interactions with either an enzyme complex or sometimes just a protein complex, like we talked about the hemoglobin example. It's not an enzyme, but it binds to oxygen. And these are molecules that have the ability to bind to one site on or subunit in a protein complex or an enzyme complex and inhibit the activity of the other subunits. So they cause a change in the rest of the complex by interacting at one site. And the converse of this would be an allosteric activator. So you can also have a molecule that binds to one site in an enzyme or a protein complex and affects the ability of the rest of the subunits in the complex to do something like either catalyze a reaction or bind to a molecule like oxygen in the case of hemoglobin. And then a sort of corollary to this is the idea of cooperativity, where we have, again, a molecule that can bind to one site in a complex and greatly change the ability of the rest of the either active sites or binding sites in that complex to do something. And very typically with cooperativity, we see that binding by a substrate to one active site in an enzyme complex causes favorable conformational changes in all the other subunits. So you see if you were plotting out the activity of an enzyme as a function of substrate concentration and you had a cooperative effect, what you would see is a very steep increase in enzyme activity as a function of very small changes in the substrate concentration because of this cooperative kind of effect. Okay? So just want to make sure that we have those terms down because when we start talking about metabolism, we're going to see that there's some interesting examples of this kind of behavior. I see a hand up. Yes. Yeah, I mean, with an allosteric activator, typically we're talking about a multi-subunit situation, okay, a multi-subunit complex. Now, in this example of cooperativity, that in your book, they talk about it as though it's sort of the same kind of thing. They show you this multi-subunit interaction, but there's lots of interesting examples of cooperative behavior in enzymes where you have a single polypeptide that shows cooperative behavior. Now, just one example of this would be, let's say you have an enzyme that has multiple metal ions that bind and do something, are required for a reaction or something like that. What you can observe is that as a function of increasing metal ion concentration, you'll see this very dramatic boost in enzyme activity, but that's not a multi-subunit situation. Whereas with allosteric reactions, typically that's what we're talking about, is a multi-subunit complex. Okay? Yes. Yes, that's right. A small change in substrate concentration will cause a large change in the rate if there's a cooperative effect. Okay, so now we're going to start getting into metabolism, and basically this is just the introduction to the idea that living cells need energy from outside sources, right? They need a food source, and what we're going to talk about in chapter 9 is how energy is basically utilized in cells to regenerate molecules of ATP that then can be used to drive the work of the cell. And so the overview of this, sort of the ecosystem, I guess, is the fact that light energy is entering the ecosystem. It's actually harvested by photosynthesis in plants that's happening inside of chloroplasts. And during photosynthesis, this light energy is used to synthesize more complex organic molecules and with the production of oxygen. And it's these molecules and oxygen that are then utilized in mitochondria to basically regenerate ATP molecules, and in the process, these organic molecules are broken down into carbon dioxide and water. And this is a constant cycle that's going on, right? So light energy enters the ecosystem, more complex molecules are synthesized, and then eventually they're broken down, and ATP is regenerated and used to power the work of the cell. And of course, this is not a perfect cycle, right? So heat is given off in the process. Yes? Well, because ATP is constantly being made and then broken down, right? It's just sort of a constant back and forth cycle. It's made and then it's broken down and rephosphorylated and rebroken down. Okay? So in all of this, in cellular respiration, we're talking about pathways that involve catabolic enzymatic reactions, in other words, the breakdown of organic molecules. And as we talked about before, these are generally exergonic reactions. That means that energy is released in the process, and it's that energy that the cell has to harvest in the form of generation of ATP molecules. So over the next few lectures, we're going to talk about fermentation. This is a partial degradation of sugars that occurs in the absence of oxygen. We're going to talk about aerobic respiration, which consumes organic molecules and oxygen and produces ATP molecules. And then we can also talk about anaerobic respiration, which is similar to aerobic respiration, but instead of using oxygen in the process, it uses other kinds of compounds other than oxygen, such as sulfur, for example. Okay, and so all of these reactions are, or virtually all of them, turn out to be redox reactions. They involve oxidation and reduction. This means the transfer of electrons during these chemical reactions. And as we're going to see, that electron transfer is a way that the cell is able to harvest some of the energy that is released during the breakdown of these more complex molecules and use it eventually to synthesize molecules of ATP. Right? And so this sort of goes back to some general chemistry. You know that chemical reactions that transfer electrons between reactants are called oxidation reduction reactions or redox reactions. During oxidation, a substance loses electrons, becomes oxidized. In reduction reactions, a substance gains electrons and becomes reduced. And overall, the amount of positive charge is reduced during that process. And the book gives a very, very simple example of this in showing you this reaction right here, in which sodium and chloride come together. In this reaction, the sodium is becoming oxidized. It loses an electron to become a sodium ion, and the chlorine gains an electron to become a chloride ion. Okay, and so that's just a very simple example of a redox reaction. And of course, the principles are similar for organic molecules as well. So when we have an electron donor, we call that the reducing agent. And conversely, if we have an electron receptor, we call it the oxidizing agent. Right? And some redox reactions don't transfer electrons, but they change the electron sharing in covalent bonds. And so we can look at it, again, this is an example from the book, is a reaction between methane and oxygen, in which we have methane reacting with oxygen, and in the process becoming oxidized to carbon dioxide. And meanwhile, the oxygen, which is the oxidizing agent in this reaction, becomes reduced to water. Okay? And in the process, energy is released. And so what these metabolic pathways have figured out how to do is to use this very general kind of chemical principle to break down more complex molecules, but in the process, harvest this energy that's released, or at least some of it, in the form of capturing it as ATP molecules ultimately. And so overall, the reaction that's going on during cellular respiration is this one right here. It's basically the breakdown of glucose molecules and the production of, ultimately, the waste products here, carbon dioxide and water. And so we can think about glucose as being really the fuel of cellular respiration. Now, of course, there's lots of other kinds of sugar molecules out there, and we talked about the fact that sugars are often stored as big polysaccharides. They can be broken down into simpler sugars that might be just 2 or 3 or 4 carbon units long, sugar units long. But ultimately, these are all broken down into glucose, or as I'll show you when we go through the actual glycolytic pathway, sometimes these simple sugars can enter the pathway at different steps downstream of the initial, where the initial glucose molecule can enter the pathway. But remember when we talked about the example of yeast being able to live on glucose, but not cellobiose, right? That 2 sugar breakdown product of cellulose. Well, the reason, in part, is that yeast don't naturally have enzymes to break down cellobiose molecules, but if we can figure out a way to introduce the enzymes to do that, such as in that example that we talked about in class, then we produce glucose molecules that can then very readily enter the glycolytic pathway in yeast. Okay, so very important to think about the fact that ultimately these pathways all have to get to glucose molecules so they can actually enter cellular respiration like this. And this just now sort of talks about the steps in the pathway. And what we're going to see is that there's this stepwise energy harvest using NAD, which we talked about last time, an important coenzyme in the cell, and what's called the electron transport chain. So cellular respiration, so we have glucose and sometimes other organic molecules being broken down in a series of steps. These electrons are usually first transferred to NAD in its oxidized form, NAD+, which is this important coenzyme. And in these reactions, the NAD+ is acting as an electron acceptor, right? It's an oxidizing agent during cellular respiration, and it becomes reduced to NADH, the reduced form. And this is, in this form, it's actually representing stored energy that is then later tapped to produce more molecules of ATP. And we won't talk about how that works today, but later this week we will discuss that in a lot of detail. Right, so this is showing the chemistry that's going on. So we have enzymes, dehydrogenase enzymes that are responsible for basically pulling hydrogen off of one molecule and using it to reduce NAD+ to NADH. So this is the very general form of the chemistry that's going on in all these reactions that we'll see, where NAD+ is being reduced to NADH. And just as a reminder, this is showing you again the structure of NAD. And remember how we talked about the fact that a lot of these coenzymes are actually built around an adenosine unit, which is shown right here. So here's the sugar of the adenosine. Here's the base. Right. So this just looks like, this part right here looks just like what you would find in an RNA molecule. There's the extra 2' hydroxyl that you find in RNA. That wouldn't be there in DNA. And then what's interesting here is that this is actually a dinucleotide. But does this look like a dinucleotide that you would find in an RNA molecule? Anybody? If I digested down a long RNA molecule to dinucleotides, would I ever get this? No. And there's a couple of reasons for that. One is that, of course, that doesn't look like one of the natural bases. But there's another important reason, too. What about that linkage? Does that look like a natural linkage in RNA? It's actually not, right? So if you look at the structure of RNA or DNA, you might notice that these molecules are connected up through their 5' carbons, through these 2 phosphates, whereas if we had a natural linkage in RNA or DNA, it's actually a linkage that involves connections between the 5' carbon of one nucleotide and the 3' carbon of the other, right? These 2 would be linked together by a phosphodiester bond. But here, we have 2 phosphates linking up the 2 5' carbons. So this is an interesting molecule because it has this fundamental unit, adenosine, that's found in RNA, but it really is a different kind of molecule, not something we would ever find actually in RNA. And in any event, so this is showing you the actual chemistry that goes on when NAD+, which is shown here, is reduced to NADH. And this is obviously a reversible reaction, right? So we can have reduction, which puts an extra proton over here. We can have oxidation, which takes it off. And so all the time, every time when we go through these metabolic reactions and you see NAD plus going to NADH or vice versa, this is actually the chemistry that's going on, okay, this kind of redox chemistry. And so what NADH does is to pass electrons into the electron transport chain. And so this is very important because unlike an uncontrolled kind of reaction, what the electron transport chain does is to pass electrons along in a series of steps so that the cell can actually make use of the energy that's being released in each of those steps. And so ultimately, this energy, as we keep saying, is used to regenerate ATP. And there's a diagram in the book that just kind of shows a comparison of the overall chemistry that's going on and how the cell is able to harvest that chemistry by having these redox reactions occur in a series of steps rather than in one overall reaction. Because, of course, we have this going on. Right? There's an explosive release of heat and light energy, like if we wanted to power a rocket or something. But it's not very useful to the cell because the energy is, first of all, would be pretty destructive to the cell. And secondly, it would be very hard to actually trap it or harvest it in some way that would lead to molecules that could then be used later for carrying out other kinds of work in the cell. So instead what happens is that the same overall chemistry occurs, but it occurs in this electron transport chain where we have electrons moving from higher energy states to lower energy states along the chain. And in the process, there's a controlled release of energy that's used to generate molecules of ATP. And these, as we said earlier, are essentially the currency. They're sort of like the energy currency of the cell. They can then be used to drive all sorts of other reactions. So it's a very convenient way for the cell to harvest energy and save it for later, or use it for all sorts of other kinds of reactions that maybe aren't directly coupled to the electron transport chain itself. Okay, so does everybody have their eye clicker? I know one person doesn't, but let's do one quick clicker question. This is kind of hopefully an easy one. And this will give you about 45 seconds to do this one, okay? And I'm going to start the clock right now. Okay, about 10 seconds. Okay, 2, 1, stop. Okay, very interesting. So we're roughly split. Okay, half the class said the answer is C. Okay, taking electrons from food and transferring them to oxygen to make water and using energy released to make ATP. That's the answer that I have down. Now, half of you said the answer was D, reducing large organic molecules to smaller organic molecules and using the energy released to make ATP. Now, I can see why you might have thought that would be the answer because you're thinking about the word reducing meaning reducing the size or breaking it down. But actually, if you think about it in a chemical sense, these molecules are actually being oxidized, right? They're not reduced in the process. They're actually oxidized in the process, releasing energy. Okay, are you happy, Mike? Yeah, he's happy. Good. All right. So that was that one. Okay, so we're going to talk now about just sort of an overview of cellular respiration. And this is a preview. And the important point here is that cellular respiration, we can break it down into 3 separate stages. The first stage is glycolysis, and this is just the breakdown of glucose into 2 3-carbon molecules that are called pyruvate. Okay, so this is a stepwise pathway that leads to the breakdown of glucose into 2 molecules of pyruvate. And those molecules then are able to enter the second stage of cellular respiration, which is the citric acid cycle. Right? So when these pyruvate molecules enter the citric acid cycle, they're then broken down further. So that completes the breakdown of glucose. And then finally, the third stage is oxidative phosphorylation. This is actually where most of the ATP synthesis is actually going on. Okay, so let's take a look at where this is actually going on. Where are these different stages of cellular respiration happening inside the cell? All right, and so right now we're talking about a eukaryotic cell, right? A cell that has a nucleus and it has mitochondria, and we start off with glycolysis happening in the cytosol, right? So this is going on in the cytosol. These enzymes are soluble enzymes, so that means they're in the cytosol. They're waiting for glucose molecules to get in, and when they do, they get broken down in a series of steps that we're going to talk about to these 3-carbon molecules called pyruvate. And in the process, a couple things happen. First of all, we get some electrons that are transferred via NADH, so we have production of these NADH molecules, and we also have the production of ATP. Okay, and this is in a process that we call substrate level phosphorylation to make ATP. And I'll tell you, I'll show you an example of what that really means in a moment. And so after this glycolysis occurs and we have breakdown to pyruvate, then those 3-carbon pyruvate molecules actually enter the mitochondria. They go across the membrane, and once they get in, they encounter enzymes that are involved in the citric acid cycle. And you'll also hear the citric acid cycle called the tricarboxylic acid cycle, TCA. Right? You might see that word, and you might have heard that before, the TCA cycle, tricarboxylic acid cycle. And that's because it's a 3-carbon molecule that's actually entering the cycle. All right, in the process of the citric acid cycle, of course, we also are able to transfer electrons via NADH and a related coenzyme that's called FADH2 in this case, in the reduced form. And we also generate molecules of ATP through substrate level phosphorylation. And then finally, in the third stage of respiration, where we're generating the bulk, about roughly 90% of the ATP that's actually made is generated during oxidative phosphorylation over here, which involves an electron transport chain and a process called chemiosmosis. And this is where, as I said, about 90% of the ATP that's made is generated over here, okay, through oxidative phosphorylation, not substrate level phosphorylation. Okay, and we'll talk about what the differences are there and why these pathways are set up the way that they are. All right, so as I said, the process that generates most of the ATP is called oxidative phosphorylation. It's powered by a whole series of redox reactions, and we get almost 90% of the ATP generated during cellular respiration is generated during oxidative phosphorylation. Okay, and then we make a smaller amount of ATP during glycolysis and the citric acid cycle through substrate level phosphorylation. So what's the difference?